So our next talk will be Neurochemical, GABA and Glutamate, Cognitive Dynamics, and Myelination and Cognitive Flexibility. And the speaker is Deepika Shukla from Nanyang Technological University in Singapore. Thank you.
So, just a minute. Good afternoon, everyone. So I'll be presenting here on today on neurochemical, cognitive, and myelination changes in cognitive flexibility. This work is a part of CLICK study that applies neuroscience to develop evidence-based programs enhancing lifelong learning and cognitive flexibility. CLICK basically stands for Center of Lifelong Learning and Individualized Cognition, which is a joint program between Nanyang Technological University and University of Cambridge. Sorry. So this one is okay. So I have no disclosures and conflict of interest. Moving forward, so let's start with what cognitive flexibility is. So in simple terms, we could say that cognitive flexibility is individuals' ability to adapt or their behavioral and thinking in response of changing environmental demands, basically in probabilistic manner. CF plays a key role in higher order executive functions like planning, reasoning, and problem solving. It also supports environmental adaptability, critical thinking, and self-regulation. CF is quite relevant in maintaining quality of life, especially through managing emotional well-being in academic, social, and outcomes, which is very much important in current fast-paced changing society, relationships, and job roles. Different interventional studies like physical and cognitive training have investigated ways to improve cognitive flexibility, but so far, no study basically investigates these trainings under uncertain or probabilistic changes. So now we mainly, this is the main part of our study. So this is structured learning, which basically helps us to extract patterns from uncertain environments, which are like similar to how we learn in real life settings. Past research has shown that structured learning training improves skills like rule inference or strategy updating, which are key to develop cognitive flexibility. Interestingly, there's another group, Feng and colleague. They have also found that the individuals who have higher cognitive flexibility, they also performed well in probabilistic tasks. This further supports our approach of using targeted structured learning training to improve or enhance cognitive flexibility in individuals. Here we intend to explore a neurocognitive mechanism which underlies basically the structured learning and cognitive flexibility with a focus on understanding how learning-induced plasticity and neurochemical modulation contributes to these processes. The dorsolateral prefrontal cortex is a key region for executive control, particularly in selecting, maintaining, and shifting its strategies or rule during the task. Although previous studies have investigated and found that DLPFC is a key region in both structured learning and cognitive flexibility, but there is a limited knowledge on how these neurochemical factors, such as inhibitory and excitatory balance and microstructural changes like myelination, they support these functions. Moreover, the potential lateralization role of DLPFC in SL and CF also remains unexplored because most of the studies either selected left or the right side of DLPFC for their investigation. But so this further restricts our understanding of how these brain actually adopts or uses the bilateral DLPFC connections in different learning contexts. Oops. Oh, yeah. Sorry, I think I didn't change the slide. Yeah. In this study, we aim to bridge existing research gap by exploring how structural learning training influences both neurochemical and microstructural changes within bilateral DLPFC region, which is quite critical in cognitive flexibility. Here, we are specifically interested in how these brain changes relates to two key aspects of cognitive flexibility, which is set shifting and strategy use. We hypothesize that structural training will alter inhibitory and excitatory balance in DLPFC within the training group, but not in the control group. Additionally, we expect that these neurochemical changes will be linked to the improvements in both SL and cognitive flexibility performance. So we adopted a multimodal neurocognitive approach to investigate both brain and behavioral changes. We had a total of 108 participants, which were divided into training and control group. We carefully matched them for their age, gender, IQ, and baseline cognitive flexibility. The neuroimaging data was administered at both pre and post settings. So we included myelination mapping, and also the MRS spectroscopy, which includes for the GABA and glutamate estimation. We have used a pseudo-randomized design with the participants assigned either to control or structural learning group. We had a total of 54 participants in each group. Only training participants or training group has undergone the structural learning training, while the control group has not been exposed to it. During structural learning training, participants will be seeing a sequence of symbols. They are basically 9 to 13 symbols, and the way they present, they have a contingency rule of the change from 80 to 20, and the participant has to predict that which would be the next upcoming signal based on their the rule formation, and there is no particular feedback was provided within these trials. So here's a brief demo on how this structural learning was. So here we see the continuous symbols are being presented, and there would be a red dot queue, and then participants have to respond on that. So this is, participants have to complete overall like minimum of 6 to overall 13 sessions on different days. Each session lasts for around 1 hour, and it consists of 5 blocks of 60 trial each. So the participants were not given any feedback within these trials, but at the end of the completion of one single block, they have been shown with the overall performance. So for these structural learning training, we have estimated 3 main key measures, which is to estimate how the performance and strategy basically evolve through the learning sessions. So to investigate or examine how these SL training transfers to cognitive flexibility, we focused on set switching and strategy components of cognitive flexibility. So for set switching, we have used a color shape task, where participants either respond to color or shape, depending on the presented cue. And by using the difference in their performance in the different repeat and shift trial, we have estimated switch cost to assess how the performance goes. And then for the strategy use, we applied intra-extra dimensional task. So for this, we have chosen only the extra dimensional task, where the participants' performance were used, mainly the extracted parameters were used to model and get a different measure called IED beta score, which basically reflects a choice determinism factor, which is that means basically high IED beta corresponds to their higher flexibility in different choice adaptation, which is quite similar what we do observe in the structural learning. And this basically corresponds to the maximization strategy in the SL learning as well. So here we are presenting a very few, because of the time constraint, we are just limiting our behavioral findings here. So we found a significant difference in the change basically the delta corresponds to the pre to post changes in the performance, and we found a significant improvement in the CST accuracy, in the training group as compared to control. But there were no significant differences found in the reaction time or the ID beta score between the group. So then we tried to see that how these SL measures basically correlated with the cognitive flexibility performance measures. So if we look at the top black box, we see that there is no significant correlation between the structural learning measures with the cognitive flexibility performance, but there has been a significant correlation between the MICD, which is basically the strategy adapted in the structural learning task, and that has a moderate positive correlation with the IED beta score, which reflects CF strategy development. So this further supports that training in the SL strategy is bit transferred or transferable to CF. For neuroimaging data analysis, we have chosen bilateral DLPFC and estimated these GABA and glutamate measures. For the myelination, we have used MPM multi-parametric maps, and we have used a standard normalized DLPFC mask from the left and right in the gray and white matter tissues. So we did not find any significant differences in other measures, so we are just highlighting here these significant components here. So we found a significant difference, or basically the decrease in change in glutamate level in the training group. And in terms of myelination, we found that the training group and control group had a significant difference in myelination in their baseline level. Then we tried to explore the relationship between the neuroimaging measures with the SL outcomes. So here in the circle, we could see the left circle, left side of part of the figure represents the imaging measures from the left DLPFC, and the right side represents the right DLPFC. Blue corresponds to MRS, and yellow corresponds to myelination measures. Here, no significant differences were found between the myelination and SL measures, but there is an interesting factor of that. We have found a significant correlation from the left MRS and imaging measures with their SL strategy. Yeah, just 2 slides. And in the right side, we have found correspondingly opposite relation with the IE balance with the mean strategy in the SL. Okay, and then we try to compare the relationship between the CF measures with the neuroimaging measures between the control and training group. So we observed that there has been a significant change in the neuroimaging measures or IE balance in the training group if we look at the improved correlation in the training group. And also the myelination is supported by inhibition. And then we found there is a significant improvement in the myelination connection, even within the local, that is in the left or right side, within the tissue, and also the contralateral. So yeah, and finally, looking at the neuro measures with the cognitive flexibility association, we found that this is bit dominant on the right side, and the training participants have found that the IE balance and the myelination are quite supportive or associated with the strategy developments in CF. So in conclusion, I would like to say that behavioral findings supports that the SL strategies through the SL training is actually transferable or supports the CF strategy development. And also the observations we found in the change in neurochemical and myelination changes suggest that there is a lateralized effect in or mechanism in the new learning. And also the key findings would be that there would have found, which is quite significant through our analysis, that there have been significant microstructural changes or tissue integrity connections between the contralateral and local sites. Yes, so this, I'd like to acknowledge our co-authors, the team, even the from both NTU and Cambridge side, our research participants, radiographer, and our imaging facilities, and also through the sponsor and funding sources. And this is a poster we have So we have Annabelle Chen here. So she is a part of a group. She is actually the leader of at NTU side. So we also have open positions for postdoctoral and research associate positions. So you can, if you like, you can connect to us. Thank you.
Time for a question.
I'm going to steal the mic for a second. Great talk. I love stimulating the DLPSC. I was wondering, do you find any session effects? I think you said you tested, got people in the fMRI scanner 13 times. Mm-hmm. Do you see any session by session effects on like myelination and GABA and glutamate, or have you not looked at that yet?
So looking at the myelination, we did not found any significant difference through sessions, but there was an effect. Mainly the significant effect was on a difference measures on the glutamate level, and we found there is a significant reduction in the glutamate that would support that basically the reduced increased inhibition basically in the right side is supporting the learning. So,
Yeah.
but myelination-wise, we did not found any session or group-wise differences.
Okay, cool. No worries. Thank you.
So I guess that it's hard. So first of all, thank you for the talk. But my question is, maybe it's a little bit hard to answer it with your data. But do you think that is going to last? I mean, you train them, you measure them. If I'll come back after a year, will it last?
I don't think so.
So
And also because all our imaging findings are basically done at the end of just the next day after the training session
Yeah.
ends. So it also now we cannot be very confident about how if we do the scan after just a week, whether the same findings would be there. But yeah, this is, I think, an investigation, or it could be was basically a part of pilot, and then we are trying to investigate more onto it.
okay.
Thank you.
Thanks to all the speakers. We did have a last speaker that was scheduled but was not present for the session. So this is the end of the session, and I wanted to thank all the speakers for their stimulating and important talks. And if you want to stick around and talk with some of them, I think they might stay for